Now the question might be, what if we don't start with a homozygous dominant and a homozygous recessive? What if we start with an individual that is homozygous dominant? So if I told you guys something was homozygous dominant, how would you represent that? Capital letter, capital letter. So if we chose A's, we could do that. And let's say we cross it this time with an individual that's heterozygous. How do you represent heterozygous? That's with a capital and a lowercase letter. So this would be the P generation. So we're going to cross them. We'll set up our Punnett square to get the F1 generation. There's a big A. There's a big A. This one could donate a big A or a little a. And then we just cross them. Big A, big A, big A, little a, big A, big A, big A, little a. So what is the genotype ratio? It's going to be 2 big A's to 2 big A little A's. Or we could reduce that and say it's 1 to 1. Or once again, we could say it's 50% big A to 50% big A little a. So that's one way we could show it. Now the other question is, what is the phenotype ratio? So the phenotype, we have big A, 2 of them. And remember that represents the dominant trait. So we could say, whatever this dominant trait happens to be, we could say it's being right-handed. Well, if we look, we have 2 of them also. So that person has the dominant trait, they're right-handed. That person has the dominant trait, they're right-handed, right-handed, right-handed. So this works out to be 100% right-handed. If we're saying right-handed is dominant over left-handed. So the phenotype ratio is the characteristic and the genotype ratio is the genetic makeup. And that was with the F1 generation. Now the question is, how do we get to the F2 generation? I won't make you guys go to the F2 generation if you start with a cross like this, because the problem is, what if you cross these two or these two? You have different options now, where if the example I gave you before, there was only one option. So right there is where we can stop on this, the F1 generation. As long as you can do the Punnett square and find the genotype-phenotype ratio, you're fine. Now we can do one more example of where we can go all the way to F2. And once again, you should be able to do this, and we'll do it as an example. All right, so let's say that we have another homozygous dominant crossed with a homozygous recessive, and this is the P generation. I want to know the F1 generation. Well, we do the same thing we've been doing. We set up our Punnett square, big A, little a, little a, big little, big little, big little, big little. And in this case, we can, sorry about that, do that again real quick. In this case, we can go to the F2 generation, because if you look at what our options are in F1 generation, they're all the same. So it doesn't matter if we pick whatever ones. We could pick those two, those two, those two, those two, those two. Doesn't matter because they're all the same. So for the F2 generation, we then have a big A, little a crossed with a big A, little a. We set up our Punnett square. Big A, little a, big A, little a. And that's big A, big A, big A, little a, big A, little a, little a, little a. So genotype, it's a 1 to 2 to 1 ratio. So 1 big A to 2 big A, little a's to 1 little a, little a. And then the phenotype, so this is for genotype. And then the phenotype ratio, they have the dominant trait, they have a dominant trait, they have a dominant trait. So we could just say 3 dominant, because I didn't define what the dominant was, 2, 1 recessive. So an example that you guys should be able to work through. But in any case, if I give you any cross, you should be able to tell me what the genotype-phenotype ratio is of the F1 generation, or in this case, we can go all the way to the F2 generation. So once again, I will hold open sessions if anything doesn't make sense, or if you're practicing, you can come and ask me or send me an email and I'll try to help you work through them. The next thing we can look at is what's called a test cross. When you look at an organism's phenotype, that does not necessarily tell you what its genotype could be. So for instance, if we go back to that purple flower idea, if you're looking at the genotype, it could be big P, or it could be big P, little p, because in both cases, that's going to give you a purple flower. So when you don't know what the genotype is and you need to figure that out, you can do something called a test cross. In a test cross, you take the unknown, so in this case, we don't know if it's big P, or big P, little p, and we cross it with a known. The only known you ever know is homozygous recessive, because if it's recessive, it has to have the little recessive alleles, and if it's homozygous, they have to be the same. So you always take the unknown, which is homozygous dominant or heterozygous, and cross it with a homozygous recessive, and that's called a test cross. So in our example of a test cross, we have our purple flower, which we don't know what it is genotype-wise. It could be big P, or big P, little p. But over here, we have a white flower that we're going to cross it with to do a test, and that's going to be homozygous recessive, little p. That's the one you always know. And whenever you have an unknown, like this over here, you always cross it with a known. Your known is always a homozygous recessive. So if we did a cross, and let's say the original parent happened to be this, well, we could get an answer for what the outcome would be. So we have our little p from that one, crossed with our big P from this one. So if we do this out, big P, little p, big P, little p, big P, little p, big P, little p. If the original parent was homozygous dominant, in a test cross, all the F1 generation would come out to be the dominant trait. They would all have purple flowers. But what happens if the original parent that's unknown was heterozygous? So let's look at that as an option crossed with the homozygous recessive. So here's our Punnett square. We have our little p crossed with a big P, little p. So we get big P, little p, big P, little p, little p, little p, little p, little p. So if your original parent was a heterozygous, that means in your test cross, half of the offspring would have the recessive trait and the other half would have the dominant trait. So by doing a test cross, you can then verify back what the original was. In a test cross, if all the offspring have the dominant trait, it had to be homozygous dominant to start with. In a test cross, if any of the offspring have a recessive trait, then it had to be heterozygous that you started with. You guys should be able to do a test cross on the exam, and once again, there'll be a homework assignment where you're going to practice these also. So everything we've been talking about so far that Mendel did has been looking at one trait at a time, like flower color, or seed shape, or plant height, and that's called a monohybrid cross. So he's crossing plants looking at mono, which means one characteristic. Next, Mendel wanted to follow two characteristics at a time. And if it followed two characteristics, it's going to be called a dihybrid cross. And the idea here is that we're crossing and looking at two characteristics together. In one of Mendel's experiments, he was looking at both the color of the seed and the texture of the seed, whether it was round or wrinkled or yellow or green. So when you have a sperm being produced from the male part of the plant, it could have the characteristic of producing yellow seeds, green seeds, round seeds, or wrinkled seeds. Same thing with the female with the egg. It could make yellow seeds, green seeds, round seeds, or wrinkled seeds. Now, if you're looking at two characteristics, remember looking at one characteristic, you can do a cross like this. But if you're looking at two characteristics together, then it's much more complicated. There's actually 16 different possible combinations that could result in this. So let's look at an example. So in this case, P generation, he had yellow round seeds, so yellow is dominant, round is dominant, crossed with green wrinkled seeds. So green is recessive and wrinkled is recessive. So all the F1 generation would have come out to be yellow round because that dominant trait would have been passed on. Now if he takes the F1 generation, the big Y, little y, big R, little r, crosses it on himself, Then you see that you have these combinations. You could have yellow smooth seeds or round seeds. You could also have yellow wrinkled seeds. You could have green round or smooth seeds, and you could have green wrinkled seeds like there. The question is, what are these ratios? Well, he found that there's a 93-3:1 ratio, but let's break this down a little bit differently. If you go back to that chart that I just showed you on the previous slide and look at just yellow, how many of them are yellow? You'll count there's 12. How many of them are green? You'll count that there's 4. Well, 12-4, that's a 3-1 ratio. And then if you go back at that chart and look and count how many of those seeds were round, there were 12. How many were wrinkled? There were 4. That's another 3-1 ratio. So even though he was looking at two traits together, yellow round, yellow wrinkled, green wrinkled, if you look at them individually, they still come out to those 3-1 ratios. So the law of independent assortment states that each pair of alleles segregate independently. In other words, when the homologous chromosomes find each other in a cell and that cell divides, they'll separate apart. So if there was a characteristic here and here, let's say that this was for yellow, this is for green, well, the yellow would be now over here and the green would now be over here. They separate independent of each other. So remember, if we look at more than one chromosome now, so let's say that we have homologous chromosomes and homologous chromosomes, and let's say that yellow, green are there, and let's say that round and wrinkled are there. Well, when these separate out into 2 cells, or eventually into 4, you're going to have the yellow here, the round here, you're going to have the green here, and the wrinkled here. So you see that they end up in different sex cells at the end of the day. And if they're in different sex cells, that means sperm or egg would carry each one of them. But since you look at them independently, you still come back to that 3-1 ratio. This only applies for genes on different chromosomes. If genes are in the same chromosome, they would be inherited together. And we'll look at that in our next chapter, not right now though. Now, last thing I'll say about this idea of a dihybrid cross. You're not going to have to work out one of those 16-box Punnett squares like you just saw in that previous slide. That's not what I'm going to make you guys do. What I want you to do is something even more simple than that. If we go back to that characteristic of, a yellow seed that's round, or let me use letters that you can see are different. So let's say we have a heterozygous for one characteristic and a heterozygous with another characteristic. What I want you guys to be able to do is tell me what would end up in the sperm cell, because these are going to separate apart and these are going to separate apart. So a sperm cell would end up with a big A and a big B, or a big A and a little b, or a little a and a big B, or a little a and a little b. Same thing with an egg. It could end up with a big A, big B, big A, little b, little a, big B, or little a, little b. And that's because they sort independently. So if you've ever taken algebra, you'll know there's something called the FOIL method. So for instance, if I go back to what I just had drawn out, you take the first, outer, inner, and last. They can come out together, they can come out together, they can come out together, or they can come out together. You should know that for the exam. If you have a dihybrid, what would the sperm or egg carry? And once again, for this example, it's big A, big B, big A, little b, little a, big B, or little a, little b. They're the 4 possible combinations you could have. That's what I want you guys to understand here. Now, here's one of the amazing things about the work Mendel did. When you look at those 4 principles he came up with that I showed you guys earlier, that was all based on that mathematical 3-1 ratio that he kept getting. Remember, when he did his work, they had no idea what a gene was. They didn't know what chromosomes were. They didn't know that DNA was the genetic makeup. So it's amazing that he was able to come up with those 4 principles that you have to know just based on that simple 3-1 ratio. However, Mendel also got lucky. Not all genes and alleles worked exactly the way he described. when he was looking at flower color and there was big P, or big P, or big P, little p, he got lucky that that color only had two different options and one was dominant over the other. Not all of our genes are like that. Some of our alleles are not completely dominant or recessive, and I'm going to give you guys examples of that. Some genes have more than two alleles, so if you look at this idea, what would happen if there's actually, this and this? What happens if there's more than two alleles? And then the other thing is that sometimes one gene can actually have multiple phenotypical effects. So there's multiple phenotypes that could come out of that one gene. So he got lucky that he didn't find any genes or any of those characteristics that fall under these, because mathematically it would not have resulted in what he found. So a lot of what he did is really genius, and some of it, like in any science, is just getting lucky sometimes. So the first term that I want you guys to know is called codominance. Codominance is when you have two alleles that both affect the phenotype in separate ways, but they're there and present together. In other words, they dominate together. One does not mask out the other one. So an example of this would be looking at what's called the MN blood group. On your red blood cells, you have these markers that stick out. And these markers are different shapes based on your antigens you produce. So an antigen is a self-marker, and it's what identifies your cells to your immune system. So your body knows what's your cells versus what's not your cells. So there are, for instance, A markers, B markers. That's what gives you the AB, ABO blood type. There's also other ones. For instance, there's ones called MN. So if you're homozygous for MM, that means you have the M antigen. If you're homozygous for NN, that means you have the N antigen. But if you're heterozygous and you have M and N, that means you have both of them on your cell surface. So let's say in this example we have ones that look like this. Let's say we have ones that look like this. So this person would be the M antigen, they would be MM. This person would be the N antigen, they would have a genotype of NN. But what would this person be? So they have an M and an N, so they're actually heterozygous. They have the genotype MN. Not the M or the N dominate, they actually are present together, so that's called codominance. It's when both alleles both present together at the same time. One does not hide the other. Now another example that you can find is called incomplete dominance. And incomplete dominance is actually where both alleles are present together, but they don't dominate together, they actually blend together. So even though the blending hypothesis isn't true, all of our genes don't blend, there are some that do. So there are certain traits that will blend together. And a great example of this is snapdragons. So when you take a red snapdragon and cross it with a white snapdragon, you actually get a pink, because the red and white combine to make pink. So if red was dominant, then the F1 should all be red. If white was dominant, then that F1 should be all white. But they both blend together, so that results in pink snapdragons. But it's not completely diluted out. You don't lose those colors, because if you take two pink ones and cross them, and the F2, you can actually get red, pink, and white flowers coming back again. So it doesn't support the blending hypothesis, but it does show that there are traits that do blend their characteristics together, but could be separated back out again. So visually, here we have the red, here we have the white. When we cross them, we get pink. But when you take two pinks and cross them, here's that Punnett square, you can actually get a red, pinks, and white again. So that's called codominance, when those characteristics blend together like this.